Please join me in welcoming to the Distinctive Voices podium, Dr. Atul Butte.
Thank you so much for that introduction. Thank you all for coming out tonight, and thanks for having me here. This is the 5th annual Seymour Benzer lecture. Seymour Benzer was later in life a chemist and really an inventor of computational technology, but actually started in genetics. And kind of ironic that I get to give this talk as well, because I'm a similar kind of hybrid, as you'll see. So thanks again for coming out tonight and for the National Academy of Science for inviting me down. First, since I'm a medical doctor, I always have to start with my disclosure slide. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You probably don't want to believe another word I say over the next hour or so. I wouldn't blame you. I really wouldn't. But I'm most proud of the bottom right here. These are all the companies started by my students. More than half my graduate students now start companies. And they do this even if they go into academia. And they do this with the most amazing platform in the world. And that's data. And it's big data. And it's open big data. I'm going to show you how they do it. I'm going to show you how I do it. And maybe I'm going to convince you that there's something in this data as well for you to get excited about. So a lot of people use a slide like this as a slide of shame. I use this as my slide of significance. If you haven't heard, we're in the middle of a data deluge. There were questions already in the front about this. The human species now generates 2 zettabytes of data every year, according to this special issue, this guy collecting ones and zeros in the rain. 2 zettabytes of data. If you forget your metric prefixes, it goes kilo, mega, giga, terabyte, petabyte, exabyte, zettabyte. And then next year, of course, we'll have 4 zettabytes of data because, of course, this number is doubling every year or two. Now, I'll be the first to admit most of these zettabytes of data are these YouTube videos of kittens playing with yarn on the piano. Perhaps entertainment value, maybe not much more than that. But I'll also be the first to admit that there are scientific data sets in these zettabytes as well. So for example, the Large Hadron Collider generated petabytes of data. Actually, it's open to the public. Petabytes of data that they use to find new subatomic particles. NASA announced two months ago that they have so many telescopes now pointing upwards that they generate an exabyte of data every day. Okay? Every day now. Now, something interesting about their exabytes, though. Okay, they even admit now, they generate so many pictures of the sky that there aren't enough astronomers in the world to look at all these pictures, okay? So it's interesting. So what do? Well, they open up the pictures to the public. They put the raw scientific data on the internet. You can register to be a citizen scientist, and they crowdsource this. Getting your friends on the internet is called crowdsourcing, right? In the old days, we used to call that Tom Sawyer getting his friends to paint the fence, right? But now we call this crowdsourcing, right? So that's an interesting theme. You're going to hear this theme again. We have so much scientific data, there aren't enough scientists in the world to even look at all this data anymore. And we're in that world today. What an amazing time it is to be in this world. And of course, we have this kind of data, and we think about this in science a lot. In fact, this article that came out of Wired magazine in 2007 was very influential to me. Some people say we have so much data now that science itself is getting obsolete. Okay? Or at least the scientific method. Now, how can science get obsolete? How can you really say that, right? Well, think about what you learned back in 3rd grade. What is the scientific method? You ask a question, right? We call it a hypothesis, and you go make measurements to answer that question or hypothesis, right? But we're already in this world that we already have the measurements. We already have the data. 99% of the work now is figuring out what's the question we want to ask, right? We have the measurements. What's the killer question here that everyone's been dying to know the answer to for 100 years, and nobody's realized we can actually ask this question now, right? So you ask anyone in my research group, in my lab, 99% of the work is figuring out what's the question you want to ask here, right? It's an interesting twist, so it's kind of backwards now. Now, people theorize that this is the next big scientific revolution, data mining, big data, think about scientific paradigms from theory to experimentation, think about computers and simulation in the 1900s, even simulated organisms like model organisms for disease. Now it's data mining. The journal Nature says it's shameful if we don't take use or make use of this data. Science says we need to make this data maximally available. And even the Lancet says if we don't start to share research data, it's going to be a problem for public health. Now, where does a lot of this data come from? It comes from all amazing new technologies in my world in biomedicine. So on the left here is an example of just one of many types of devices and tools we have in biomedicine to measure a lot of molecules. On the left here this is called an Affymetrix gene chip. It kind of looks fancy. This is what one of these actually looks like if you've ever seen one. I always bring a prop with me on talks because data is really, it feels ephemeral, right? You never see data. It actually comes from amazing gizmos like this, okay? Data comes from small packages, big data from small packages, right? And this is just one of many types of measurements. I mean, look, we're in a Beckman building, right? So Beckman Coulter had sorting cells and counting and looking at their cell surfaces. That was the original one of these a long time ago, right? In the 70s. And now we have even more recent technologies with DNA sequencing. So we have a lot of technologies here. This one is the size of your thumbnail. Here's a thumbnail for comparison. And this happens to quantitate. This quantitates every gene in the genome, measures every gene in the genome. This one looks at the levels. You can also look at the spelling of these recipes, but we're looking at the levels, how much is being cooked on the cells. You can put a sample of diabetes or cancer, normal tissue, a mouse, or for man, and you could just get a digital readout of all the genes. Then when you get tired of measuring them one by one, here's the 96-well plate version, right? Some of you might have ever spent time in a lab. We love these 96-well plates because it's a multiple of 12 by 8. And each well now is one of these chips, okay? Now, this is an amazing gadget, right? It kind of even looks like a computer chip by design because Affymetrix is in Santa Clara, that's right next door to Intel, so they make it look like a computer chip. As amazing as this is, though, this is already a 15-year-old technology. We've had this already for 15 years, okay? This might be the first time you're seeing this. We've had this for 15 years. Everyone runs these today. Every university, every major teaching hospital, medical center runs these already. And what's amazing about this technology isn't this technology. What happened is in the early 2000s, 2001, 2002, everyone started writing papers, research papers on this kind of data. Here's a list of genes that are changed in that cancer. Here's a list of genes that are changing in Alzheimer's disease or diabetes. And they submitted all these papers to the journals. And what did the journal editors say? They said, hold on a second. We're getting way too many of these kinds of papers. No more papers with these gene chips. No more papers. Unless you show me the data, okay? Put that data into an international repository so other people can kind of double check the math. And maybe someday people can use those samples for something else. So this was a paper from Charles Perot back in 2001. Many calls for data sharing at that point because reviewers of papers and the public should see this raw data. So what's the most amazing part of this chip isn't even the chip. The most amazing part is we somehow can make scientists share this raw data on the internet today. Fast forward, so this is 2001. Fast forward to August of 2012, and we hit a milestone. This was in Nature, it featured my lab and others. We hit 1 million of these chips publicly available. Okay? Not just the 60 I started my career with 15 years ago. There's 1 million of them, right? Actually, 2012 was forever ago. If you look today, there's already 1.7 million of these samples publicly available. Here's one website, here's the other. This is run by NIH. This is run by the Europeans, the European Bioinformatics Institute over in Hingston, UK. Doesn't matter which one you get to, it's still about doubling every 3 years. In this hour, we will probably add another 100, right? Just by doing nothing. 100 samples just now newly available to the public. Now, it's easy to get lost in the millions and billions, so let me just make this really concrete for you. What exactly does this mean? There's a search box in the middle of this website here, right? Some of you are scientists. Any of you, forget about you. A high school kid who needs to do a science fair project, right? 11th grade. She can go to this website, type in breast cancer, and find and download 50,000 samples of breast cancer already run, already digitized, about as easily as finding a song on iTunes today, right? 50,000 samples of breast cancer already publicly available. Now let's just think about that for a second. What's so magical about 50,000 samples, right? That's more samples that this high school kid has than any breast cancer researcher, right? Because by definition, any one breast cancer researcher has to put their data there, and the one kid who knows how to get all the data out of the repository now has more than any one researcher, right? Now, 50,000 samples isn't enough for you. Just wait 2 years, it'll be 100,000 samples, right? Now, actually, the interesting number here is this number 2,000. 2,000 independent labs, independent groups have already run something to do with breast cancer, cell lines or tumor biopsies or dog models or humans or mouse models. All that data is just public available. 2,000 labs have contributed data. Let me tell you how amazingly crazy that is. Let's say I'm a brand new researcher in breast cancer, brand new researcher, and I'm writing a grant proposal. You're all the judges for my grant proposal, okay? And I propose to you, I'm a brand new researcher in breast cancer. I don't know anyone in this field. But in my proposal to you, I propose that I'll go get the 2,000 best labs in the world studying breast cancer to run their latest technologies available on their most characterized patients and give me all that data for free, okay? Now, if I propose this to you in a forward direction, this sounds ridiculous. Why would 2,000 labs even say hello to me as a new researcher in breast cancer, right? How can I even get friends on the internet to help me? 2,000 of these labs. Yet here it is. 2,000 labs are helping you. So when I think of public data like this, I call it not just crowdsourcing, retroactive crowdsourcing. 2,000 labs are there on the internet to help you, and they don't even know they're helping you. Actually, 2,000 labs are there on the internet to help you, and you don't even know they're helping you. And for some reason, if that's not enough for you, just wait another couple years, there'll be 4,000 labs. What an amazing world this is. Whether it's breast cancer, colon cancer, prostate cancer, all of these labs get one paper out of their data set, and then they release all the data waiting for you. Because there's nobody else out there to do this right now. It's just sitting there waiting for you. Now, if you have any doubts a high school kid can do it, here's the high school kid who did it, okay? 17-year-old, this is Brittany Wenger. She grew up in Sarasota, Florida. She's now a Duke undergrad. I've yet to meet her. I've mentioned her in 100 talks. Someday I should meet her. And she won the top prize, first prize in the Google Science Fair competition that year. She's got the white trophy there because she built an artificial brain. What that means is a neural network to diagnose breast cancer. She took publicly available breast cancer RNA data sets and some pictures, immunohistochemistry, and she figured out a way to come up with a diagnostic. And then she was bored, I guess, the next year, so she was in the front page of all the newspapers for doing it again with leukemia. So of course what I mean, though, is if a high school kid can do this, all of our scientists today should be able to do this today. Yet you know where I'm going. We don't train scientists at all in computational tools or to use this data at all. And think about why we don't. Why is it like that? Where the data just keeps coming. If those 1.7, 1.8 million samples are enough for you, if you're a cancer researcher, boy, something called the Cancer Genome Atlas that just got finished last year, 11,000 samples of cancer from 20 different kinds of cancers. And here they're looking at the DNA and the RNA and the protein measurements, even the mammography pictures and the brain scans. All that data released to the public. There are no names on these or identifiers, so you're not going to be able to tell which patient was which, of course. Got to keep that anonymous. All that data just sitting there waiting for someone to do something with this. dbGaP. If you're a genetics researcher now, funded by the National Institutes of Health, NIH, if you're an NIH-funded genetics researcher, boy, it's not the journals that make you share data now, it's the funding agency. NIH says, if you don't start to share data to the public, we're not going to give you any more money, okay? Even as fast as next year. So it starts with the journals, and now it's the funding agency. Now, here's an example. So genetics data actually we're pretty finicky about, right? Because genetics, you've watched, Law and Order or any of these kind of crime shows. You know we can identify people with DNA. So here we're a little bit more picky, right? Here, you can't just be a high school kid. You have to go get IRB approval, ethics board approval, to show you're an actual scientist and you can download data. It's not hard, but you can do that. But here also, NIH makes you share the data even before you've written a paper about it, okay? Even before you get the chance to write your manuscript, you got to put the data out there on the internet, okay? Well, to be fair to those scientists, we have these embargo dates. That means we got to give them some time to write a paper. You're allowed to get the data, but you can't write your own paper until after the embargo date. Boy, you look at this website, so many of these already expired, 2007, 2008. Anytime you go to this website, more than half the data set's already expired. Well, why is it worth all that hassle? Why am I even bothering to show you this particular slide here, right? At the top, all the way in tiny, tiny lettering here, is something called the Framingham Heart Study. Anyone remember that? It's why we know the word cholesterol in the United States, right? Framingham's a town just about 15 miles west of Boston. They've been studied for, it turns out, 60 years now. And the actual residents of Framingham and their descendants and some of their descendants have been just followed forever, 70 years. Now, it's kind of interesting. You can go and finally download the genetics on 14,000 people. But actually, it's even more amazing to me. You can get 70 years of measurements on these people, right? Inflammatory measurements and clinical measurements and heights and weights and who lived next door to whom and who was friends with whom. All that data publicly available today. You could even go back and just say, well, let me just look for everyone that's had, I don't know, pick a disease, lupus. Let's say everyone that's had lupus in 2010. Okay, now, are there any markers that we could have told who was going to get lupus 10 years earlier, 5 years earlier? Maybe we can start to predict who's going to get which disease. And you know, the answer is just sitting in that data set. And if you think they've written every last paper they can possibly write in 70 years, you know you're wrong. It's so much left to discover. And I'm just showing you that Framingham is one of 10,000 studies on this list, waiting for you. Nobody else is there to do this. When a group gets a data set collected, you know what the next thing they do is? They write a paper and they go get money to get another data set collected. So few people just keep looking at this data. What an amazing world. My favorite is this one, PubChem. We're kind of in a chemistry building, so I'll put this one out here. PubChem is an interesting, what happened about 15 years ago, Elias Zerhouni took over NIH, and he was kind of dismayed. He saw the pharmaceutical companies, of course, making a lot of money. We'll talk about pharmaceutical companies. That's what they do. But they weren't targeting rare diseases and orphan diseases. So he said we should give some money to academics to do this. And so what's exactly involved is you could come up with a biological model, some cells, that maybe cancer cells. Let's say they're glowing if they're living. And then just test 1,000 drugs or 1,000,000 drugs or 1,000,000 chemicals. It's called drug screening. Get a robot to just test one after another, just see if the glowing gets better or gets worse. But if NIH funded you to do that drug screening, you got to deposit that data into PubChem. What is PubChem? PubChem, think of it as a grid. 170 million drugs are all future drugs as the columns. 1.1 million of these drug screening things as the rows. If you do that math, it's 190 trillion little boxes, of which 1 billion of them have some measurement made by some poor soul in the past 10 years of this drug in this assay, okay? If you know anything about drugs or chemistries, 122 million of them meet Lipinski's rule of 5. That means it could be really drugs in the future. They can get absorbed just right. 1 million of the billion were designated as active. This one actually worked in that screen. Why am I showing you this one? I can easily bet you a beer this beats any pharmaceutical company. Ah, hell, I can bet you a bottle of wine. This beats all pharmaceutical companies combined, right? Because this is doubling every 3 years, and very little is doubling anymore in the pharma discovery world. You can see layoffs here and there, right? And they give this away without a user ID and password. Probably every future drug is a column in this grid somewhere, waiting for you. Just sitting there, waiting for someone to do something with this data. What an amazing time we're living in right now. I know there's a million reasons to be negative about the world and NIH funding, and then there's this. They give you all this away, ready for you. So let me just tell you what we do with this kind of data, right? So my first big grant at Stanford, I was at Stanford for 10 years. I just moved to UCSF. I'll talk about that at the end. My first big grant was to go after, why pick one disease? Go get every human disease studied with these chips. Just go get them all. Cancer, diabetes, childhood diseases, aging disease. Just go get every human disease. And we collect them. We look for diseases or experiments where someone looked at diabetes or disease samples and healthy. Disease and healthy. Disease and healthy. We collect these kinds of experiments. Now, it turns out it's actually not so hard to figure out if they're disease samples in an experiment. It's actually really hard to tell if they were healthy samples. There are 200 words for healthy, it turns out, right? Normal, vehicle, wild type, time zero. The surgeons use a word called margins. That's the part outside, right? And of course, the word control, right? We even had to write a paper on the 200 words people use for normal, okay? Because we have to use text mining and computational tools to read all these experiments, and we start to capture them all. So actually, though, if you pick any one disease, you actually don't have one experiment, you have hundreds of experiments, right? I showed you the breast cancer, you got 2,000 people who've already studied this. And so what we do in my lab, in my group, is we don't trust any one of these data sets. I love all scientists, but I don't trust any one of these experiments. But I trust what's in common across all of them, right? If, I don't know, 50% of the researchers are seeing the same genes up or down or the same results, maybe I can believe that. And maybe if they're all seeing the same thing going up or down, the same thing again and again, maybe it's actually not just a bystander, maybe it's part of the real process. It's causal in the process here. That's the kind of thinking here. Now, we were sitting on all this data, and then we realized, I'm a medical doctor. I realized, what's the first thing we're going to use this data for? Well, we need to come up with more blood tests for people, what are called diagnostics, okay? We need more diagnostics. For example, we don't have a good blood test for breast cancer, right? We have mammography, which has a lot of problems. We really don't have a good test for ovarian cancer, right? It just happens, and then we're just kind of lost, because we don't have any good blood tests. So we figured, let's use some of this public data for blood tests, for making blood tests. Now, it's always kind of dangerous to show the central dogma in an evening audience, but let's just try here. This is the central dogma in molecular biology. This is it. If you know anything, if you want to remember anything, just remember this. DNA goes to RNA, goes to protein. Now, this is a little bit for aficionados for a second. What happens is a cell, you're probably kind of maybe getting hungry, so your body wants some sugar. It's upregulating some proteins. And the cell says, I want to make this protein. It unravels the DNA, and it makes a blueprint and sends the blueprint out. And for the aficionados in the audience, these chips measure the blueprints. We wish we could measure the proteins, but measuring proteins like this is still a very hard problem. A lot of chemistry involved. The RNA is more of a digital readout. But you know what? RNA doesn't make a really good blood test. Okay? Why is that? I spent 1 year in the wet biology lab. That was a really dangerous year. But I learned one thing in that lab here. If you just look at RNA the wrong way, it starts to disappear on you. Did you know that? Right? If you just look at it funny, the RNA starts to degrade. Because your body gives off RNases, things to degrade. Your body likes a bubble of no RNA around it. Because RNA to your body means it's a virus. So let's just create this bubble of RNA. So RNA is really tough to work with. Protein is really easy to work with. Think about the last time you saw a screaming child with a fever and a sore throat, and somehow a pediatrician like me sticks a Q-tip down there, and they get enough to figure, oh yeah, this is strep, right? This is a strep throat. How did that happen? That's kind of that's 1970s proteomics. That's called an ELISA. You can get a little bit of protein, and you see a big signal. Protein measurements work in a desert. RNA barely works in a lab. So we said, well, we want blood tests for proteins, but all we're measuring are the RNA on these chips, and it's really kind of as simple as we think. If the RNA is up, let's go test the protein. There's a little arrow there in between. That's as simple as that. Fine, so we get a lot of ideas now. We turn our crank on our computer, we get a lot of ideas. Here's a blood test for that, and here's a blood test for this, and we said, here's a blood test for acute myeloid leukemia, AML, okay? This is not a good leukemia to get, AML. They're talking about putting someone's bone marrow into you when they say you have this disease. So we say, okay, well, look, we turned our crank. We got a new diagnostic for AML. Maybe it'll tell us who's going to get AML in the future or relapse. That's great. Okay, so what do? Well, we got to test these proteins, this computational prediction. So we go to the Stanford Cancer Center. I love my cancer center. I'm a member of the cancer center. I said, do you have any saved blood from patients with AML? They said, you know what? We saved all the RNA, but we never saved any of that liquid around, that blood, the serum or the plasma, okay? It takes more freezers, it turns out, and they decided not to do that. They said, okay, we're Stanford, right? You go get the IRB approval, put up those posters you see in the big hospitals to join my study. You should have enough samples in a year. And what does a computational person like me do? We just Google for it. Instead, we go to a company like Conversant Bio. Oh, I see pictures of blood tubes. I'm in the right store. Browse by disease. What do you want to buy today? Bladder cancer, brain cancer, breast cancer. Here's leukemia. Click there. Now, what exactly did you want to buy? CD19 positive B cells from leukemia, CD45 bone marrow cells, leukemia, T cell negative selection leukemia, bone marrow. Page 2, oh, here's plasma from peripheral blood leukemia. $55 each. How many samples do you want per patient? Add to shopping cart. What the hell is this, right? This is real. I'm not making this up, right? It turns out big hospitals in the United States and all over the world, when you get a blood test ordered, if you're a doctor, you order a blood test, or a nurse practitioner, the lab saved that blood for about 48 hours, thinking the doctor might add something else to be run on that sample, right? They save it for about 48 hours. But at the end of 48 hours, they got to make room in the fridge, right? You know, it's got to get thrown out. And something magically legal happens on the way to the trash, okay? If they take off that sticker and put, we don't know who this patient is anymore. We put on a new sticker that says which disease and which sample. Companies come in, organize this, and resell this for research use. Now, I, as a customer, have to agree not to try to figure out who these people are. I don't truly care who these people are. But this sample shows up in 72 hours on dry ice, ready for you. This is not the hard part anymore. If you have an idea for a blood test, especially in the cancer world, here's your sample right then and there, okay? It just shows up. Now, what an amazing world, right? You think about, Now, I said one point. I said it's magically legal. I didn't say it's ethical, okay? There's a difference. Ethics are region-by-region decision. For example, they get away with a whole lot of stuff in Boston we can't get away with in Palo Alto, let me tell you. Or even in California, because we care for our patients to know what's happening to their samples, right? We inform you. Other parts, the community just says, you're big hospitals, you guys do great stuff, go use our samples, right? So ethics are region-by-region thing. I'm not saying this is ethically everywhere, but it's legal in the United States to use discarded samples like this. And this is how it's done. If you want samples, pathology samples, let's say you're so moved by, Steve Jobs dying of pancreatic cancer. Well, now you can buy 100 samples of pancreatic cancer. Well, actually 200, because there were 42 cases of this kind of cancer, 3 patients had that kind, 1 islet cell, 6 metastatic, some tumors, some pancreatitis, 2 samples of each. So all of these patients went to the OR, they had a hunk of tissue taken out, and after it's been read, it's going to go in the trash. They stick in new needles, they get samples, they put them on what's called a tissue microarray, $395 each, a little picture of a shopping cart. Shows up. This one doesn't even show up on dry ice because it's already been fixed, right? 48 hours later, you're ready to go to test your idea. You're not waiting years anymore. But you got to trust these companies. We'll talk about trust in a little bit. So we start to turn our crank. We said, boy, we can do a lot of these. We had one nice paper out on cancer paint. What's a cancer paint? It turns out when kids get bad brain cancers like medulloblastoma, the surgeons go in, they got to take out this cancer, otherwise the kid is going to die. But they got to leave as much brain as possible for this kid to grow up with, right? They cannot really tell the difference in the OR. They said, we need a paint. Well, how do you design a paint that you can paint the operating field and then it glows, in the dark to see, where's the cancer? Well, you got to figure out what's in the cancer, what's not in normal tissue. So literally, we go to public data, we figure out what's in medulloblastoma, subtract out normal neurons, astrocytes, glial cells, dendritic cells, subtract, and then you figure out, well, here's our paint. This is what's left. And you can see this is darker than the other one. You don't even need to know what the markers are. It doesn't matter. Did everything online, did everything with samples we bought off the internet. Now we got this paint that we're designing. We did this for transplant rejection. It turns out when patients get a new organ put in, right, like a heart, a lung, or kidney, some mornings you wake up and the body says, hold on a second, this isn't part of me. I got to get rid of this thing. It's called acute rejection. Body starts to attack that organ. It's not painful. You don't know this is happening. So a doctor has to keep sticking needles in to see if it's happening. We said, that's ridiculous. That's archaic. We need a blood test for this. So go get every public data set with these kinds of rejections being studied, heart and lung and kidney and all the rest, and just figure out what's in common. And with that, we came up with a brand new marker, kidney, liver, and heart. We said, why should we have a separate one for heart and a different one for kidney? Let's make a universal blood test for rejection. And we got one of those too. But my favorite one is this one, preeclampsia. Okay, this one hit me close to home. My wife had this. My daughter was born really early because of it. She's fine now. Preeclampsia. Has anyone heard of preeclampsia? A couple hands here, right? In the United States, the only reason most people have heard of preeclampsia is because one of the major characters in Downton Abbey dies of this disease, right? Spoiler alert. I'm sorry if you haven't watched the show yet. That's the only reason we in America have even heard of this usually, or maybe you know friends who've had this. This is still how husbands lose wives around the world. The blood pressure shoots up, and actually you got to do something. Now this disease unfortunately falls right in between two fields of medicine. The obstetricians, I love them, but their job is kind of simple. They say the delivery is the cure. Take the baby out, the mom is better in 48 hours, right? All the blood pressure comes down. But then we pediatricians have to deal with this baby for 10 weeks trying to keep them alive and healthy, right? It falls right between the cracks. And there was no blood test for this. Forget about blood tests like to tell if it's coming. There's no blood test to even know if it's happening right now because of all the complications, other things that can cause blood pressure to go up. So Linda Liu, a grad student in my lab, Bruce Ling did the validation, Matt Cooper is involved in more of the science. We said, we got to fix this. So what do we do? Well, first we go to the public data, literally. If you just go to the repository and say, let's just show me all data sets with preterm birth and preeclampsia, 266 to choose from. And just pick and choose. Well, we want these larger experiments, forget about the smaller ones, figure out what's in common and chase it down. And that becomes a blood test. So here's an example of blood test. Hemopexin is up in preeclampsia compared to normal. Now here, you see a paper, fine. Now here you see a company logo, because this becomes a company now. And that's another point I want to really stress here, right? I'm going to show you a couple arrows like this. We start with the unmet need, public data available. We get some internal funding from the March of Dimes. They fund premature birth research. We have that at Stanford, and we continue that at UCSF. Data is analyzed. We design a diagnostic. We get another internal grant to test some of the actual protein measurements, $50,000. Spin out a company, $2 million in private seed financing. I'm not showing you $2 million to brag about it. I'm showing you $2 million because this is the new way science is going to get done out of academia. Can I tell you how hard it is to get a brand new NIH grant for $2 million today? You know it's hard. So the point is to move it closer to patients. The science continues in the company. It's not all simple in the company either. I got to figure out what is this hemopexin and all these other measurements. So how do we actually make it closer to a real product to help patients and families and healthcare providers? And so this is the new thinking for me, is that if we want to change the world, we can't just keep writing papers about it. We have to actually file some intellectual property, see if someone's going to license this, and you know what? If they don't, we got to start the companies ourselves. And that's where I'm showing you this. Of course, we're creating jobs along the way and all of that goodness, but the point is new science is funded by on the private side into a company. This may work or not work, but we got to do more of these. And it's all enabled because we had the data to start with here. So that's the diagnostic side. Let's talk about the drug side. Okay? How do we make something to do something about diseases like drugs? If I asked any of you how much does it cost to develop a drug, it's amazing. Most lay people know it costs $1 billion and it takes 10 years to develop a drug. Because the lay Americans don't know much in common, but they know this number because it's actually advertised on the Super Bowl, right? The pharma industry does kind of communicate, this is why our drugs cost so much. It's because it costs $1 billion and it takes 10 years to develop a drug. Actually, $1 billion is the wrong number, okay? Actually, how much does it cost to develop a drug? Well, the math is really simple. Pharmaceutical research and development spending, R&D dollars, divide by the number of drugs, right? Simple math. R&D divide by the number of drugs. And if you do that math, it actually costs between $4 billion and $12 billion to develop a new drug today. Kind of not sustainable, okay? Now, it's kind of interesting, right? So why is it getting worse? It's because the spending is going up and we're not getting that many new drugs to compensate today. It's kind of funny if you look at this list, if you play with the stock market at all. This company, Pfizer, at $7 billion, is trying to buy this company, AstraZeneca, at $12 billion. I'm not a strategic consultant, but if I were in the middle here, I'd try to buy one that can do it cheaper. I don't know why they're trying to buy that one. That's all pipeline and politics and things. That's just not sustainable. Now, why is this actually going to be a problem, okay? So few drugs are coming out, and many drugs are going off patent. They're becoming generic. Now, don't get me wrong. When drugs come off patent, it is good for all of us, okay? It's good when my patients can afford the drug, right? That's the point. That's a good thing for society. But so few new drugs are coming out that by 2018, the industry is predicting the entire pharma industry will lose $250 billion of revenue that year. Okay? I didn't say they're going to make $250 million less. They're going to make $250 billion less dollars in 2018. Now, let me be clear, right? You may hate the pharma industry on this side. You may love the pharma industry on that side. I am not here to pass judgment on the pharma industry here, but I'm kind of worried if they lose $250 billion of revenue, who's going to pay for the new drugs that are costing $12 billion each? And then you realize it's all going to have to come down to us to do this. And by us, I don't just mean us at large institutions like UCSF and UC Irvine. I mean even smaller companies, even just working off a couple folks in a lab. It's going to come down to us to have to come up with these drugs because they kind of don't know what's happening here. And I'm worried, and we need more drugs, especially for diseases where nobody's addressing these drugs. So, as we were collecting experiments with disease and healthy, we start to realize we can use public data on drugs. I already showed you that big grid called PubChem. Well, people, investigators all over the world, especially in Boston and elsewhere, the Broad Institute, they take cells and they put with drug, without drug, with drug, without drug. So on the left, you have with disease, without disease, and on the right, you got with drug and without drug. So the story on this, the reporters called this Match.com for drugs, okay? Now, some of you are laughing, so you know what Match.com is. This is how younger folks find, spouses, dates, significant others. And what's the famous saying? Opposites attract, right? Where opposites attract. So if now these colors just represent the genes going up or down, doesn't really matter what the numbers are. I'll just show you the statistics with my arms. If I've got a disease where this gene went up and this gene went down, and over on the right I can find a drug that can make this gene go down and this gene go up, maybe there's a match there, right? All I'm looking for naively is a drug that can reverse the effect of the disease. I'm seeing all measured on the same chips, right? Which genes went up and down? Let's just see if we can reverse them. Some of the mathematics folks in the audience can imagine correlation coefficients and things like that. I'm not going to show you any of that. You could just reverse it. This is with 2 genes. Imagine with 10 or 20,000 genes. That's how many genes there are in the genome. So you get the idea here. Now we start to turn our crank, and we start to get ideas. Boy, this chemical could work on this disease, and this drug could work on that disease. But where we have the most fun and interest is not a new drug, but finding a new use for an old drug. The idea here is called drug repositioning or drug repurposing. And why go after those? Because we're already using them in patients. They're good for something, maybe they're good for something else. Now, there are many famous examples of drug repositioning. We didn't invent this idea. One of the early examples was a cardiac drug that had an interesting side effect of hair growth. That's today minoxidil. Maybe some of you use that for your baldness. But of course, everyone knows the other cardiac drug that had an interesting trial. Nobody wants to release the drug after the trial. That today is Viagra, right? That is Viagra, started as a cardiac for angina drug, right? People thought it could open up blood vessels in the heart. Viagra started as a cardiac drug. Now, actually, people don't realize Viagra has done a double reposition. There's another drug called Revatio. It's the same molecule, but that's the name you're supposed to use to open up the blood vessels in babies from the heart to the lungs. It's called pulmonary hypertension. Those blood vessels get too tiny, and this just opens them up. It opens up those blood vessels, too. And there were all these jokes about babies on Viagra at the time, but it kind of works for that. But here is the point, right? Instead of finding these new uses by accident, how about we find them on purpose? Using public data to do this, right? So I got data on the drugs, data on diseases, and we're just going to match them up. So we start to turn our crank, and we start to get ideas on this, right? And then we start to go to our friends. Well, you've got patients with this. Can you test this drug? Before you put it into patients. We need to put this into mouse models, right? You can't just go try drugs in patients. You got to test them in mouse models of this disease or that disease. So we go around, well, we have a couple friends here on that disease or this disease, and you run out of friends really quickly, right? Because they think you're moving too fast as this computer. Why should they try anything? So now, when I really, really need a drug tested in a mouse model, I go to assaydepot.com. Kind of sounds like Home Depot, and you're right, okay? These are my aisles: biology, chemistry, DMPK is drug metabolism, pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology and toxicology. Okay, let's click on pharmacology. What type of mouse do you want to run today? Bone, cardiovascular, dermatology, diabetes, urinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. I'm a diabetes guy. Let's just click on that. Okay, here, look, I found it. Here's a 16 mouse study. Maybe I have 2 groups of 8 or 4 groups of 4, I don't know. OB diabetes model. If you read here, it says this mouse has been eating a lot and getting diabetes since 1951. This is a standard model for how mice get diabetes. It's not the only one, but it's one of the original ones. It goes way back. And in 28 days, they'll give you all the fasting blood sugar, and they'll run an insulin tolerance test and glucose tolerance test. Some of you might have had that when you were pregnant. They do all those tests in the mice, and they'll see if this drug worked or not, okay? And you don't have to tell them the name of the drug because you send everything blinded. They don't have to know anything. What am I covering up here? The price. $9,000 for this service, 9-week turnaround time, and literally add to shopping cart. You can buy an entire mouse model off the internet today with a credit card today. This is here and now. What an amazing world. Now, the scientists that run mouse models in their labs, let me be clear, right? There are plenty at UC Irvine and my campus. They look at this price, $9,000 for a 16-mouse experiment? That is ridiculously expensive because they have slaves, graduate students, that can do this work for much cheaper, right? Of course. But for dry bench scientists like me that don't want to go in that animal facility, this is, what does MasterCard say? This is priceless, right? This is priceless to get actual data if this works. Now, some of you are asking yourself, what is he talking about? You have to trust this company, right? Before you put this into humans? This seems like the most important part of your experiment. Forget all that computer stuff. How do you trust this company here? Well, first of all, some of you with sharp eyes will see this is run in China. Some of you may have a problem with that, others not. I don't. But in reality, when you hit add to shopping cart, you're presented with all 133 companies that are willing to offer this service to you, including Maryland and Wisconsin, okay? So outsourcing doesn't always mean offshoring, okay? Maybe you just want the USDA-approved ones. Well, click here. There's only 4 of them. FDA, 5 of them. ISO 9001, 7. You click here for certifications, and you're only going to see the ones that are so certified. Oh, how I wish it were this easy to pick the right collaborators on my campus, right? So I knew some random undergrad wasn't doing the mouse experiment, but someone with some certifications, right? Still, though, you're wondering, how do you trust this company? Everything depends on this experiment. And I'll tell you, if you really, really need to know, like we do, whether this drug works in this mouse model, simply order 2 of them from 2 companies. Hell, at $9,000, order 10 of them from 10 companies. Why do I say that? Think about the mouse researcher today on any campus. When they get their mouse experiment working, do they go across the hall to get a completely independent team of researchers to reproduce their experiment? You know they don't do that. So what I've suddenly done is I've converted the quality problem as a function of dollars now. You want even better quality, get more dollars, right? It's only $9,000 each. Get 100 companies to do this for you. And the quality problem completely disappears because I can just recruit as many companies as there are out there to do my experiment for me, right? Quality is not an issue anymore. So we start to do this. We start to turn our crank. We start to get ideas. Our first big idea was a seizure drug, topiramate, we predicted would work on inflammatory bowel disease. It's called Crohn's disease or ulcerative colitis. And here you can see this is what a normal rat colon looks like. You add this chemical, it gets red and inflamed. You give steroids. You can use steroids. You can't keep patients on that forever, but it suppresses the inflammation. And you give this seizure drug, topiramate. This also just works as fine. Now, this is an interesting experiment, right? We get this data. Our good friends, Mohan Chano, Jay Pashrika, they run all these experiments for us. We submit this to Science, Translational Medicine. An editor kicks us back and says, we love this data. We can see the drug is working. But do you have any better data on this? Because our good friends gave us iPhone pictures of the colon, okay, of the intestines, right? God, we have great microscopes on campus. And they give us iPhone pictures here. And I said, Mohan and Jay, we love you. We want this paper really published. We're going to outsource this. So we go to Assay Depot, and we just find 2 companies. We find one out in Berkeley that was having a sale, okay? For the same price as everyone else, they'll run all the immune cytokines like TNF-alpha, other immune cytokines that were useful to have, and they saved all the tissue for us. And then we picked one company out in Cambridge, Massachusetts, freaking expensive, but the only ones in the world who knew how to do a rat colonoscopy. Think about those 2 words together, okay? You know you've had colonoscopies. Rat colonoscopy. These are my famous rat colonoscopy videos. This is a normal rat colon. Here's one with that chemical giving it redness. And here's one that's all better with the seizure drug. And we got 40 megs of video on the website to show you that this drug actually works. Now, a couple interesting points here, right? Nobody at Stanford knew how to do a rat colonoscopy, right? So I'm not just outsourcing to save money, I'm outsourcing to get the best damn data in the world. A company knows how to do this? Why would I learn how to do this if I can actually figure this out? I'm just going to buy this, right? So it's not just to save money, it's to get the best data out there, right? And so this works. We've done like a dozen of these. This is my favorite one now. This one, those came out a couple years ago. This one came out about 2 years ago. Now this is about cancer. Now, this is a worse disease, small cell lung cancer. This is a really bad cancer. Humans get this from smoking. If you knock out these 3 genes in a mouse, it gets its own lung cancer. This isn't a mouse where they put in human cancers. This mouse gets its own lung cancer. You see all the nodules here, okay? Now, small cell lung cancer is a bad cancer to have. 5% survival rate in 5 years. Only 5% survive. This mouse is the same kind of mouse as this one, treated with this antidepressant, imipramine, okay? We still have this antidepressant in the pharmacies, but we don't use it anymore because it's an old tricyclic antidepressant. We use serotonin reuptake inhibitors like Prozac and the rest. This one had some annoying side effects, okay? This one, in some people, made them really sleepy, and in others, it might set them up for an arrhythmia. Actually, neither of those 2 side effects sound as bad as having lung cancer. 5% survival rate. And the cancer is all melted away. It's gone. Okay? Now, I love this story. Why do I show you this one out of the dozen we've done? Because we start with public data. We get this to work. We get the prediction. We test cell lines in a dish. I'm not even going to show you that. We get this amazing mouse data. And 15 months after the computer prediction, we get the IRB, the ethics board approval, and we launch a clinical trial on this drug. 15 months from computer to patient getting dosed. We don't know if this drug is going to work or not. This one may fail. There'll be others. Total cost of the trial, $50,000. Not even $1 million here, right? We got to do more of these, okay? This one may fail. The next 10 may fail. But this is what I'm talking about. It's getting more of these predictions with data into patients here. Now, we've got to start a company on this if we're going to scale, right? So let's just show the arrow again. We have this need for these drugs, public data available. We got NIH funding. We collect all these experiments, get this idea together. We launch a company on this, get that shovel-ready era grant when they were trying to stimulate, create some jobs, hire people, and $3.5 million in Series A financing for venture capitalists. The science continues in a company. My lab goes in any direction. The science keeps going in a company to get it to patients that much sooner. Now, actually, this is why I'm in the business, though. I get a sad email like this, still one a week. Tuesday, October 8, 2013. Hello, my name is, I'm an Italian man. I live in Sweden, 48 years old. I have 3 children, 6 years old, 8 years old, 17-year-old. All with the same wife. On June 5th, this is October, June 5th, they told me I have small cell lung cancer. I work 75%. I got different metastases already, liver, kidney, pancreas, skeletal. I know I will not live longer than a few months, maybe a year. Wants to be part of this trial, right? 2013, it's probably already gone. This is why we're in the business. We need more drugs. We need them tested faster. And I refuse to believe it has to cost $1 billion to do it. And we're giving away this data, and we need more people to do this. And we got to get more kids trained, more undergrads trained, more scientists trained to use this data to get things into the clinic faster. Because I get an email like this every week, and the data is just sitting there. Now, that's a kind of serious story here, so let's lighten it up a little bit here, right? And in the end, I've talked all about molecules. But big data And life-changing data is going to come from more than just molecules. How many of you have a wearable gadget with you here? Any of you? There's only one hand here. Fitbit, Withings, okay, a couple of hands here. This is a new field, okay? The Apple Watch, Samsung Watch. Data isn't just going to come from our molecules anymore. It's going to come from our bodies as well here, right? This is a field called quantified self, okay? Now, this started, of course, in California, like all things, right? And the idea here was to measure everything going in and out of your body. Now, I can imagine measuring everything that comes into your body. Going out sounds kind of gross. But the idea here is to measure everything about yourself, your activity, how much you weigh. I used to kind of laugh at these people until December 2012. Came back from a trip from Hawaii. I stepped on a scale. I was 247 pounds. I said to myself, when I hit 250, they can weigh me in tons, right? That's like a quarter of a metric ton. What the hell happened to me, right? Well, life got really sweet at Stanford, right? And I bought everything Fitbit makes. In the last 2.25 years, I lost 50 pounds with everything they sell. One of these gadgets to measure my steps, so I try to get 10,000 steps a day. I got 8,300. By the time I'm done in LAX tonight, I'll hit 10,000, let me tell you. And I weigh myself every day on a scale. And I record everything I eat, and that way my weight doesn't deviate too far or too fast off of track. I've learned that actually you can use this, if you're a data person, to actually change your life. Now, here's an interesting concept, though. I did the most amazing intervention to change my life, to improve my health, right, to lose 50 pounds. But my doctor is the last person to hear about this because their super expensive electronic health record system doesn't even talk to my Fitbit today, right? Can you smell a business opportunity here? 10 companies working on this problem, right? Why is the doctor the last to hear about this? The data's sitting in different puddles and pools and lakes, and they got to connect these things, right? This is going to change people. It's going to change behaviors. I'm a big believer in these. Here's another company I'm impressed with. I have no commercial connection to them. Theranos. They got this massive building on Page Mill Road in Palo Alto. Super expensive. Got this one of the richest billionaires, right? One of the youngest billionaires who started Stanford dropout. This is measuring every blood test with just a finger stick. Of course, if you have diabetes, you know you can measure your sugar levels. This is measuring everything else, right? It's kind of neat gadget. I've had it done myself. If you go to the Palo Alto Walgreens on University Ave, it's the one and only place where this is actually deployed to actually test it out. Well, so here's the concept of how little blood you need. And they say, you know what, since we're going to do a new type of blood test, let's make it pretty for you. Let's put up orchids, right? Did the place you went last time get a blood test have orchids? Probably not. You know what, since we're making a new experience, let's put it at Walgreens, okay? Walgreens now will have our blood data, not UC Irvine, not UCSF, not Stanford. Our hospitals will have to talk to Walgreens and soon Walmart to get health data out, right? Now, since they're all out there in the wellness centers at Walgreens, they actually put the price out there. This is the most disruptive thing ever. CBC, complete blood count with diff, $5.35. My latte costs more than that at Starbucks, right? I know we're probably charging $80, $90, I don't know, $100 at my hospital, right? They just put the prices out there. Is this going to change the world? Of course it's going to change the world, right? $5 for a blood test and shows up on your iPhone when the results are back. But now we hospitals are going to have to talk to new providers, including Apple and Fitbit and Walmart and Walgreens, to get all our health data. In some ways, if you don't have all that data, you don't really have your patient anymore, right? They're just lost to you. Medical devices used to take 5-10 years to develop. Here's a neat medical device, like a medical tricorder. Some of you might remember Star Trek, where they wave that kind of thing over you. Well, these guys wanted to make a brand new one of these, where it measures your temperature, your heart rate, your pulse ox, your oxygen levels, your heart rate data, pulse wave. You can even pee on the thing. I don't know if I would actually pee on it, but you can do a urinalysis. It measures your stress levels. And how do you launch a brand new medical device today? You put it on Indiegogo, right? They said, well, we'll actually go and build this thing if we can raise $200,000 or $100,000 in a month. They raised $1.6 million in a weekend. People like me, I already pre-purchased one of these 2 years ago by paying $200. You can pre-order, they see the demand, and now they know they should build one of these, right? What an amazing world that medical devices can get crowdfunded today. There's the charging and the cost data as well. This was a funny story. I was giving grand rounds at the University of Miami when this showed up on the front page of the Washington Post. University of Miami charges double what Jackson Memorial does across the street. Okay? Heart attack, $166,000, $89,000. We in the health world better get this data under our belts faster than the reporters do, because the reporters know how to analyze this data. They have access to all of this. And nobody wants to be in the middle anymore. Everyone wants to be on the lower side of this. But then University of Miami can say, well, of course we charge double. Our quality is better. Not when the quality data is public too. You can't argue that either, right? See where we're going? It's all there. And of course, all this shows up in apps. You want to buy a medicine today? GoodRx. Well, let's say you need your lipid drug, Zocor. Well, CVS next to you has $197. Here, it's a coupon, $197.85 with free coupon. But why are you buying that? You should just buy the generic, right? You could just stay, here's a generic. Simvastatin. CVS with coupon, $10.75, Walgreens, $10. You can stand in the middle of a city, just spin around, and the compass will tell you which way to go for the meds, right? Hospital safety. I made this slide when I was giving a talk in San Antonio. Here I was, and I was thinking, if I had crushing chest pain right now giving this talk, right? I'm having a heart attack. Do I want to go to the B and C hospitals next to me or the A hospital down here? Probably depends on traffic, right? Oh, there's no traffic? Take me there, ambulance, take me there, right? It's an app. This is real right now, right? My favorite is Clean Hospital Finder. They ask patients, did you see someone clean your bathroom in your hospital room today? Not all the B and C ones do, but that A one does. I can guarantee you that one. Take me there. Ambulance, take me there. Now the FDA regulates apps. Did you know this? 200 apps are under FDA regulation today. Now, they're not these apps. These are apps that talk to devices, like the heart, the EKG in your iPhone case, and some of those. But can you imagine a doctor writing a prescription for an app someday? Not too far. They're already regulating 200 of these, right? What's the next big open data for me? I'm not going to spend a lot of time on this, but I run this website, clinical trials data. I put half my lab on this. What's a clinical trial? It's the reason the drugs cost $1 billion. What's a clinical trial? The most expensive experiments in the world, right? These drug companies run these experiments. When they fail, which most of them do, they don't even write a paper about it, right? So nobody learns from the failures. Forget about releasing data. It's going to change. JAMA, New England Journal saying you better release your data. It starts with the journals. And now the European version of the FDA is saying you got to release your raw data. FDA might get there someday. I see a lot of potential from all this clinical trials. For example, why do you think this drug failed? Pharmaceutical company seems to work in these people. Here's a marker for the people it works in. Let's save that drug. Let's figure out how to use it in the patients it's actually working in, patient subsetting, and all that I think we can do from the lay public. And so many more data sets. Electronic health records and microbiomes and epigenomes and side effect data and entire countries getting sequenced and all of this. How do I put it all together? I'm going to end with this concept here. To me, the future of medicine is about chronic disease. You know what chronic disease is, right? The acute care stuff is all going away. We need to follow our patients for the rest of their life. What is the job of a doctor to make sure every patient ends up with a good outcome? Well, here's a patient that just went off track and is heading towards a poor outcome. And we only get to see our patients once in a while in biomedical moments. Believe it or not, we doctors don't live with our patients yet, right? We only get to see them. Exam room, emergency room, once in a while. Maybe they're getting closer with mobile gadgets, but we still don't get to see patients all the time. The entire world is focused on what in the genome and proteome is going to change this outcome. Keep them heading towards a good outcome, not just a poor outcome. Let's throw the whole genome. But let me tell you something. You can get an entire genome on a patient, but if they cannot afford the meds, it doesn't really matter, right? What in the social context and environmental context alters this outcome? And I'll tell you another thing. You can get an entire genome on a patient and you can get a system finally where they can afford all the meds. But if we can't practice quality care, if we keep making errors and mistakes, it's all for naught, right? What in the care we're delivering is altering this outcome? Three major silos. Quality hospital people don't talk to geneticists, don't talk to the sociologists. They all have to talk together. And this is an amazing world because there's so much data in all of those. And this is what I call data-driven precision medicine. Learning from every patient in real time to figure out what can we do for every single disease moving forward. I'm going to end with this last concept because we talked about companies. In my part of California, we're so used to these kind of annoying kids that start amazing high-value companies in the garages and dorm rooms, right? Like Apple and HP. And we got dorm room companies like Yahoo, Google, and Facebook. I think when we think about precision medicine, we think about the kids, we think about the data. I think the next Genentech, the next Amgen, the next Pfizer, it's going to start in your garage. When you go home today, in your garage, you already have almost 2 million of these chips and every mouse model in the world on your credit card. I want kids today to design their cancer drugs, not another app to share kind of naked pictures with each other. We need more cancer drugs. And when I mean making drugs in your garage, I don't mean making meth in your garage, okay? I mean, we need drugs for a lot of conditions. You have all the data in the world, it's just waiting for you. And I always show a picture of my daughter who learned how to play with DNA at age 5. That's what we got to do, is get kids be just ready in the science museums learning how to do this, even when they're just ready to even think about writing and all of that. I'm now moving to UCSF. I get to run something called the Institute for Computational Health Sciences. As great as UCSF is, I actually have a position at all 5 University of California medical schools: Irvine, Davis, San Francisco, San Diego, and Los Angeles. Why do I have that position? It turns out that we can build an electronic data warehouse to share and learn from all 13 million patients seen at a UC hospital. That's 4% of the US population is seen at the University of California. We're going to build a system, we're going to learn about it, we're going to give the data back to patients, and we're going to learn how to do the right thing for patients, cost effectively, but delivering equitable care, reducing disparities, the right thing in the future. Can't do any of the work I showed you. A long list of collaborators here. I have to thank endowment support from the Lucille Packard Foundation for Children's Health. My lab has 17 NIH grants from these 9 institutes of NIH. These 4 give me more. These 5 give me less. I still love them. March of Dimes, HP, HHMI, Stem Cell, Disease-Specific Foundations. I always thank my admin and tech staff. I would never get a grant or paper out the door without them. And I always thank my wife, molecular biologist, biochemist. She reads all these big papers and grants that come out of my life. She starts companies with me and lets me go all over the world to give talks like this. Thank you very much.